Hello lovelies, in this video the brilliant Dr. Edwards is going to be taking you through our Edexcel 2023 predictions for Biology Paper 1, and this is for Combined Science Foundation. Now Dr. Edwards is going to be taking you through exactly the sort of things you should be putting in your answer, any clues you can pick up from the question to help you work out exactly what the examiners are looking for, and then how you can structure it and lots of exam tips for you.
Hello everyone. Okay, so this video is a walkthrough paper for the Edexcel separate Foundation Paper 1. So we'll be going through each question step by step. I'll analyse the questions and give you some exam tips for how you can read these questions like this in the exam and figure out what they're asking and try and link some of the content that you need to answer the questions to that answer. Give the model answer and then we'll go through the mark schemes together and I'll explain what you do get marks for, what you don't get marks for, and sort of why that is. So hopefully you've had a go at the paper already, and then you're going to go through this paper with me to mark it, or you've marked it and you just want some clarity on the mark schemes and try and figure out where you may have gone wrong. Okay, so let's start. Okay, so this question is about bacteria. Select the process by which bacteria divide. Tick one box only. So it's asking you how bacteria divide, so it's not mitosis, and you can see that's not even one of the options on the list. But the way bacteria divide is literally to split into two, and splitting is covered by the word fission. So split into two, fission, so we know, hopefully we should know, it's binary fission. But if you weren't sure, how could you kind of work out from this multiple choice which one it was? Well, I always say go through the multiple choice and think about what that word means to you, or if you know what that word is, kind of give yourself a little note next to it and then see which one helps you answer the question. So for meiosis, it's not that because that's making gametes, so that's making sperm and eggs. For phagocytosis, that's engulfing bacteria and destroying them. That's what white blood cells do. And plasmids is just the name of the loops of DNA that you can find in bacteria. So it can't be any of those. So if you know what any of those words mean, you can cross them off if you know that that's not what's going to give you the right answer. And we know that the right answer is therefore binary fission because that's what's left. You can also use your knowledge from physics, the idea of fission, splitting the atom. That's what that word comes from as well. Okay, so part B. Use the image below, figure 1, to help you describe two key differences between bacterial and animal cells. So there's a lot of things here. We're talking about two things. Key things means main or important things. So kind of really big, obvious things. And we're looking at the difference between bacteria and animal cells. This is important. It means we're not comparing bacterial cells to plant cells. If it had said compare bacteria cells and eukaryotic cells, that's different because then that includes plant cells, and there's some things that bacteria and plant cells have in common that animal cells don't. Cell wall, for example. So we're looking to try and write down some things that bacteria have but animal cells don't. So this idea of a comparative statement. So this thing has it and this thing doesn't have it. And you need to write it like that in order to get these marks. First of all, let's look at what it wants us to look at in terms of the first difference. So describe how the DNA is different in animal cells and bacterial cells. So the main, remember, it's main or important, the key thing about bacterial and animal cells in terms of DNA is there's no nucleus. There's no nucleus in this picture that you can see. There's no nucleus in bacteria. They don't have one. So now we have to just write that as a comparative statement. So if the bacteria doesn't have a nucleus, where is the DNA? You can kind of talk about it. So DNA in animal cells is inside the nucleus, but bacterial cells don't have a nucleus, so the DNA is just in the cytoplasm, which we can see in that picture. So I've said bacterial cells don't have a nucleus, animal cells do. In animal cells, the DNA is inside the nucleus. In bacterial cells, the DNA is just inside the cytoplasm. So that's my main key difference about the DNA in animal cells and bacterial cells. There are some other things you can say, and we'll look at those in the mark scheme as well in a second. Okay, so our second difference, describe how the outer structures of bacteria and animal cells differ. So things that are on the outside, around the edge, not inside in the cytoplasm this time. So it's just directing you to come to some of the structures you can see in the picture. So what can we see in the picture? Well, we can see a flagellum. Not all bacteria have a flagellum. Some do, but because they've given you one in the picture and they've asked you to use the image to help you, then you can be safe to use it in this circumstance. And also we've got a cell wall. And remember we said we're talking about animal cells, not plant cells, so it is a difference. So we need to do a comparative statement again. Bacterial cells have a cell wall, but animal cells do not. Or you can talk about the flagellum, and again, we'll look at that when we look at the mark scheme. Okay, so part C. What type of microscope would have been used to obtain the details shown in figure 1, giving reason for your answer? Okay, so remind ourselves what level of detail we can actually see in figure 1. So obviously it's a bacterial cell, and we can see the ribosomes inside the cell, and we can see the DNA inside the cell. And obviously it's a cross-section as well. All of these things should hopefully lend us evidence that it should be an electron microscope that took this picture, or we were able to draw this image from an image from an electron microscope because we can see inside structures. So an electron microscope, because internal structures, including ribosomes, can be seen, and these are too small to be seen with a light microscope. Okay, so let's have a look at the mark scheme for question 1. So 1A, as we said, is binary fission. 1BI, this and the second question, BII, we need to make sure that we're writing what we call comparative statements. That means they need to be in the correct pairs, and you need both parts to get the mark. So in animal cells, the DNA is in the nucleus, and bacterial cell DNA is not contained in the nucleus. So you're showing a really clear comparison there. In animal cells, this is what happens. In bacterial cells, this is what happens. You're showing a clear difference. And you're talking about DNA in both parts. Or bacterial cells contain DNA in plasmids and animal cells do not. Both of those are focusing on the DNA, and both of those are talking about a comparative statement. For BII, same thing. To get 2 marks, you need to have both parts. So bacterial cells can have a flagella and animal cells do not. Most bacterial cells have a cell wall and animal cells do not. You've got a clear comparison, a clear difference, and you need both of those statements as a pair to get the marks. So you could, if you just said bacterial cells have a flagella, you can have 1 mark. But if you didn't clearly say that the animal cells don't, then you don't get 2. Lastly, for 1C, it's an electron microscope. You get 1 mark for getting the right type of microscope. And then we've got different ways we can justify it, but our explanation needs to either be about the fact that there's structures inside that you wouldn't be able to see with a light microscope because they are too small. The main thing here is to say too small, or because an electron microscope has a high enough magnification and resolution to see the internal structures. So that's kind of flipping it and saying why it has to be an electron microscope because it's really good. Then the other thing that you could do if you didn't say either of those, so talk about the structures being too small to see with a light microscope and talk about what you can see in the diagram, or talking about why it has to be an electron microscope because of its power, you could talk about the opposite and say it can't be a light microscope because a light microscope has too low resolution and magnification to show that level of detail, which is similar to the first part, but you're focusing on the microscope as opposed to focusing on the structures in the cells. Any of those would get you that second mark though. Okay, so next question. Features of plants have been changed over many years as farmers have bred them for specific characteristics. An example is broccoli, which is the result of hundreds of years of breeding wild cabbage plants. So we've got a picture of some wild cabbage and its Latin name Brassica oleracea, which was discovered or what it looked like 1,000 years ago. And then we've got broccoli, which it looks like today. So it's the same species, but obviously a different variety. So this is clearly selective breeding. We've got an arrow that tells us it's selective breeding, but even without that picture, we should be able to tell that that's what this question is talking to us about. So it says the parts of the broccoli that we eat are large flower buds and thick stems. Original wild cabbage plants had small flower buds, thin stems, and large leaves. The table below shows the stages of selective breeding the farmers used to produce modern broccoli from the wild cabbage. Complete the table to show the correct order of the stages. So we've got the stages and we just need to put numbers next to them to show which is the correct order. So we've got cabbage plants are continuously bred over many generations until all plants have the characteristics. Bred the wild cabbage plants with desired characteristics together. Choose wild cabbage plants with thick stems and wild cabbage plants with large flower buds. choose offspring from the parents with the largest buds and the thickest stems. So if we're thinking about any selective breeding question, we need to kind of think of the stages. So it is select, as in select the plants or the animals with the characteristics you want, breed them together, select the offspring with the characteristics you want, and breed them together and repeat. So that's basically what we're looking for here. So choose wild cabbage plants with thick stems and wild cabbage plants with large flower buds. So that's the first thing you have to do. Select them, then breed them together, then choose from the offspring that have the large buds and the thick stems, and then continuously repeat that over generations. So hopefully we can see that the one that says select or choose the original cabbage plants should be the first one. Then we need to breed them together, which is the one above. Then we need to choose the offspring this time, so that is number 3, and then repeating over many generations is number 4. Golden rice is an example of a plant that has been genetically engineered to reduce vitamin A. It's helped to prevent vitamin A deficiencies in children where their diet consists mainly of rice, as it's hard to grow other crops. What is genetic engineering? So again, this is just a definition. Hopefully we've learnt this definition, and remember, it's not necessarily about changing genes, it's just about taking genes from one organism and transferring them to another. Okay, next part. So give 2 reasons why some people might be against genetically modified crop plants. So you may have learnt this as disadvantages of GM crops, or kind of just objections that people have to doing genetically modified crop production. But basically you can pick any 2 of the reasons that you hopefully would have learned about, things that people don't like or dislike about genetically modified crop plants. So the clue is here as well, it's crops, so this idea that it's plants that we're going to eat. So, but it could be the same as if you're talking about any other kind of genetically modified plant that you're going to grow in the wild. Genes could spread to the wild, so they obviously sexual reproduction could happen between these plants and other plants nearby, and then these new genes that we've put in could spread into other plants. The genes that we put in could interact with other genes in the plants. People do just tend to disagree with the idea of genetic modification, that it's tampering, that it's interfering with nature, and they don't like that idea. If the genes spread to the wild, they could affect food chains. There's loads of reasons. We'll look at all of them in the marks in a minute, but I'm just going to put that they could spread to the wild, and then if they do that, they could affect food chains. Okay, so let's have a look at the marks. So we've got our table there. 4, 2, 1, 3 is the order we need. If you got any one of those numbers correct in the row that it's in the table answer though, then you can have 1 mark. If you got any 2 numbers correct, you can have 2 marks. And if you got all 4 correct, then you can have 3 marks. Got that definition of genetic engineering. You need to use that definition because that should be the wording that you've learned. And then you've got any 2 from these possible bullet points, and there's extra in the except column as well. So any 2 from possible harm when eaten by humans or animals, because obviously eating this, we don't know the effects of it. Damage to food chains, that the gene may spread to other wild plant species or wild populations. Not sure how the genes are going to interact with other genes. Genetic engineering is unethical or unnatural. You could allow allergic reactions for possible harm when eaten. So if you've said about possible harm when eaten or possible allergic reactions when eaten, that would count as the same mark. Allow reduced biodiversity. So if at any point, so damage to food chains, or you've said something about it could reduce biodiversity, it could cause other species to die, then that's where you get that mark from as well. So loads of options there, you just needed 2 of any of those. Coronary heart disease is a non-communicable disease that kills 17 million people each year. State the 3 ways in which cardiovascular diseases can be treated. Okay, so we need to say 3 things. It wants 3 ways that we can treat cardiovascular disease. Now, there need to be 3 different things. So coronary heart disease is sometimes referred to as CHD. You might have learnt it or remember it as this. Non-communicable disease, remember that's a disease that can't be spread. So again, this is kind of what I would be highlighting and writing around my question. It's 3 marks. We need to say 3 things. So they've got to be 3 different things. We should have learned, hopefully there's 3 different kind of groups or types of treatment for cardiovascular disease. One of them is drugs. So statins is an example, which lower cholesterol. That's an example of drugs that can be used to treat it. You've got surgery. So there's lots of different surgeries. We could have a valve replacement. It could be a mechanical valve. We could be looking at a whole heart transplant. There's different surgical things. Maybe a stent, for example, as well, could be under surgery. So either drugs, some type of surgical replacement, or also lifestyle changes. So people may be told if they've got early signs of CHD to go on a low-fat diet to try and reduce their cholesterol. Also exercise to try and remove excess fat and things from the body. So these are kind of our 3 categories. So we can either just say drugs, surgery, lifestyle, but that's not really going to be enough. We need to kind of I'd say give examples, or at least explain a little bit sort of what you mean by lifestyle, because it needs to be very specific that you're saying that there's a change. So changing to a low-fat diet, going on a low-fat diet, or changing lifestyle, or changing to a diet and exercise regime. And then I've chosen statins as my drugs, and then I've chosen valve replacement surgery. But I've put the word surgery in there so they know what I mean by that. Okay, so part B, define the term non-communicable. Well, we've already written that as a little note off of non-communicable disease at the start, so that's helpful. And this is why I would always say annotate kind of what thoughts come into your head as you're reading the questions, because then they can help you later on. So a non-communicable disease is a disease that cannot be spread between people, or from person to person, or between organisms. Doesn't have to be between people, because diseases can spread from animals to people as well. And obviously plants diseases exist. So one person to another is fine, but organisms is fine too. Okay, part C. The following graph shows the trends in deaths from coronary heart disease. So we've got a graph, we've got men and women on the graph, we've got to describe the change that can be seen from 2000 to 2018. So describing a graph, you always just say what you see. Say what you can see from the graph. Changes is a word that's used here. So we're looking at increases or decreases over time, changes over time. And it's from 2000 to 2018. So they want the whole graph. They want to talk about the whole graph, not just a section of it, because sometimes they just want a section. And we need to use some data, really, to help us with making these points, because we should always say what we see, but try and include some data if we can. So what can we see? Let's have a look at some things that we can see to talk about. Okay, so we have an increase and then a decrease. So that's something we can talk about. That's a change. So between 2000 and 2018, we have an increase until 2003, and then we have a decrease. And then if you think about it, what we often like to do as well for describe questions, I would always say, is always mention the overall trend. So the overall trend here from the start to the finish is decrease. Although we have a slight increase and then a decrease, overall, if you look at the values at the start, so in 2000, so kind of like 1,300 up to 1,550, something like that. And then that decrease overall, we get to about less than 1,000 by 2018. So we overall have a decrease. So that's a couple of things we can already say. Increase, then decrease. And we've got some data points. So 2003 was the peak. So 2000 to 2003 increase, and then a decrease until 2018. And then the actual numbers of that overall decrease. So from about 1,300, over 1,300 to below 1,000. And so that gives us some data to talk about as well. And that's going to be given up. That's enough for 2 marks here. There's some other things that you can talk about, and we'll look at that in the mark scheme. But for me, I think that would be what I would go with for my answer. So overall, the number of deaths from CHD have decreased from over 1,300 in 2000 to less than 1,000 in 2018. And there was an increase to 2003, and then the numbers steadily decreased. Okay, so let's have a look at the mark scheme for question 3 then. So as we said, we've got medication or drugs, surgical procedures, and lifestyle changes for A. And we've got named examples allowed. So statins, artificial valves, stents, transplants, reduced fat diet, regular exercise. But for that last one, an idea of change must be implied. So as I said, you can't just put lifestyle, you need to make sure that you're saying that there's a change involved there. So because their lifestyle could be causing their CHD, they'd have to change it in a way that's going to help improve it. Cannot be passed from one person to another. As I said, organisms is allowed here. Doesn't have to be person, because obviously, as we said, communicable diseases aren't just for humans. And then we've got lots of things here we can get 2 marks for the second or the third part of the question. So correct use of scale from the y-axis anywhere in your answer. So if you've quoted a data point from the y-axis anywhere, you can get a mark, or use of any data from the graph. So if you talked about the peak, if you talked about when it peaked, if you talked about from the lowest point to the highest point, the increase, any data to back up a point gets you 1 mark. If you didn't talk about data though, you can still get marks. So deaths in men are always higher than that for women. So that's one of our kind of overall trends that we spotted. And then increases and then decreases gradually over time. So just describing the increase and then decrease. And you could have used data to help you with that point. And then another overall trend, it's an overall downwards trend, or overall the number of deaths is decreasing. However you said that, that's also a mark as well. Any 2 of those gets you 2 marks. Okay, so on to question 4. So chlamydia is a disease that is transmitted during sex. It can cause yellow or green discharge and pain. So that's the type of pathogen that leads to chlamydia. So we've got options here. Think about, when you've looked through this list, what examples have you learned for each of these? So protist will be malaria, virus will be probably HIV, and fungi is normally athlete's foot for humans. So bacteria is our only other option if you didn't remember that chlamydia was caused by bacteria. On to part B. So give one method that can prevent a person from being infected with chlamydia. Well, there's a clue at the start of the question where it says that it's transmitted during sex. So if you didn't remember that that's how chlamydia was spread, then this is going to help you. So one way to prevent the spread, if it's a sexually transmitted infection, there's only one method that you will have learned, which will be condoms, or a barrier method of contraception, such as condoms. So chlamydia can be treated with antibiotics. Explain why antibiotics cannot be used to cure viral infections. So we need to give reasons why antibiotics wouldn't work if you were infected with a virus instead of a bacteria. So the main thing is to think about, well, how do viruses work? So viruses infect cells and they replicate inside the cells, and then they burst out, and that's what causes the damage. Bacteria don't go inside your cells, they are just inside your body and inside your tissues between the cells, and they secrete the toxins which damage the cells from the outside. So if you have got a drug like antibiotics which kills bacteria, that's not going to need to go inside the cells, because the bacteria are all outside the cells. So that's why it won't kill a virus, because the virus are hiding inside the cells and the drug works outside of cells. It will never go in and attack a virus. So that's all we need to explain for 2 marks. So viruses live and replicate inside cells, and antibiotics work outside of cells. That's it. So patients with chlamydia can also be told they can take painkillers. Explain why they need to take both antibiotics and painkillers to treat the gonorrhea. So the question is just saying, give reasons why they have to take both. So we know that the antibiotics are going to kill the bacteria. So what do the painkillers do? The painkillers are going to treat the symptoms. It said at the start of the question, obviously, one of the symptoms of gonorrhea is pain. And we know that painkillers treat that. But if they just took painkillers, why would that not treat the gonorrhea? Why do they have to take both? It's because the painkillers only treat the symptoms of the infection, and they will not kill the bacteria. You need the antibiotics to kill the bacteria. So that's our answer. Okay, so painkillers only treat the symptoms of the infection. They will not kill the bacteria, but the antibiotics will. So aspirin is an example of a painkiller. Where does the chemical used in the drug aspirin originate from? So this is one of those examples of drugs that you need to know. So aspirin, the main chemical that is used in aspirin comes from willow bark, so the bark of willow trees. It's along with things like digitalis, which we need to know comes from foxgloves, and penicillin, which comes from the penicillium mold. So it's one of those 3 examples of kind of drugs that were discovered in plants or animals or organisms. New drugs are being developed for use in the future. State 3 things that new drugs need to be tested for. So these we should just, it's one of those things we need to know. We need to know these 3 reasons that we test drugs. So efficacy, which just means do they work? Dosage, which means how much is needed to work. And then toxicity, which is do they cause side effects or are they dangerous in any way to the cells in the body? So efficacy, dosage, and toxicity. And that's what we test for when we are testing new drugs in a clinical trial. Okay, let's have a look at the mark scheme for question 4. So most of those first few were fairly straightforward, 1 markers. For 4CI, it said, so the second mark, we need to talk about that human cells aren't really affected and viruses live inside human cells. So you can say that antibiotics would have to kill human cells in order to kill the viruses, as long as you've implied that viruses live inside the cells and that's not where the antibiotics work. That gets you marks here. For 4CII, you've got the 2 points, so that the painkillers only treat the symptoms, the kind of only here, or this idea of treating the symptoms and not killing the pathogen or the bacteria are the 2 points you need. So painkillers do this, they only treat symptoms, or they only treat pain, or they only treat the symptoms of the infection. They do not kill the pathogen, kill the bacteria that's causing the issue. That's the main thing there. Willow bark, you can say willow trees, that's fine. You really just need the word willow, don't need the word bark or trees, but if you put either of those, that's fine. And then for 4D, we literally just could state those words because it said state, and those are the 3 things we're looking for. So literally just those 3 bullet points gets you those 3 marks. So on to question 5, which is a practical question. So 2 students wanted to compare their reaction time using a ruler drop test. Hopefully we recognise this as one of the required practicals. There's not very many for paper 2, so this is one of them. They used the following method. Student A held the ruler just above student B's hand. Student A let go of the ruler without warning, and student B had to catch it with their thumb as soon as possible. The reading on the ruler where the student's thumb was in centimetres was recorded to show how far the ruler fell before it was caught. They repeated this 5 more times and then swapped over so student A was catching. So first of all, I'm going to note that they didn't say which hand they used. That might come in handy later. Hopefully we will remember that one of the things that you might have done if you did this at school is that you would use your non-dominant hand so that it was slightly fairer. They repeated it, which is good. Any repeating means that we don't have to talk about the fact they didn't repeat it and they only did it once. They repeated it 5 times. So the first question we've got says, complete the sentence below using an answer from the box. The distance the ruler dropped was the what variable. So we've got control, dependent, and independent. What we've been given in part 3 of the method is that they measured in centimetres on the ruler where the thumb kind of caught the ruler. So that's what they were measuring. I always try and make sure people remember it this way. So dairy milk ice cream, to remember DMIC, and then D is for dependent, M is for measure. So the dependent variable is what you measure, then I is for independent, and C is for change. So what you change in the experiment is your independent variable. So dependent is measure, independent is change. And then the control, hopefully we know we keep that the same. So the distance that was dropped was measured by the students, so that means it's the dependent variable. Obviously what they changed here was the fact that they had 2 different students. So the independent variable was the students being compared about how fast they caught the ruler. Suggest one factor the student should have controlled to make this investigation fair. So we've said control is things that you need to keep the same. So control variables, remember, are things that you need to keep the same that can affect the experiment. So in this case, they did repeat it. In this case, we don't really have any other details about things that are kept the same. So we need to think about what factors can affect reaction time. And so therefore, what things should they try to have kept the same? So the sex of the person, the age of the person, how tired they are, how much sleep they've had, for example. All of these are examples of things that can affect reaction time. There's lots. So remember, control is something you keep the same, but it's something that you keep the same because if you didn't, it would affect the experiment. Okay, so we'll look at some of the other options in the mark scheme in a minute. There's lots. I'm going to put age as my example for my answer. Okay, so now we can see the results. After converting their ruler measurements to reaction times using a table, hopefully you remember doing this, so obviously the actual distance isn't your result, you need to convert that to the reaction time in seconds. So they've got the following results. They want us to calculate the mean reaction time for student A. Now this is a 2-mark answer. So first of all, how do we calculate the mean? Well, we add up all the numbers and divide by the number of values. However, if you're calculating mean, you should know that you should do that, but only do that once you've removed any anomalies. So we need to take out any anomalous results and then add up all the numbers and divide them by the number of values. So I'm looking at the list for student A. So we've got 0.27, 0.4, 0.23, 0.24, and 0.22. Now the 0.4 is jumping out at me immediately as a potential anomaly because it's nearly double all of the other values. And if you look over to student B as well, their highest was 0.31. So it's way higher than any number on the table, whether it's for student A or student B. So I would say that this is most likely an anomaly, given how close in range all of the other numbers are. They're all in the 20s. So I would say we remove that, and so then we calculate 0.27 plus 0.23 plus 0.24 plus 0.22. That gives us a total of 0.96. And then we're dividing it by 4 because we only had 4 values, so that gives us 0.24. Okay, part D. Give one conclusion you can make from these results. Well, if you're looking for conclusions, especially in the table, then you need to look for patterns or differences that you can talk about. So there's a pattern that I can see, which is that for both students, the reaction time decreases as they have more goes. So it decreases with practice, which might be something that you looked at when you did your reaction time. And also we have a difference. So student A's mean reaction time is lower than student B, so they have a faster reaction time than student B. So there's a difference and there's a pattern. I'm going to go for the difference because that's kind of what they were investigating, was they were comparing their reaction time. Okay, so let's have a look at the mark scheme for question 5. So you've got a lot of options for 5B. Anyone from the hand that was used, the same sex of the students, the same age of the students, same time of day and amount of sleep, kind of linked, but the idea is obviously they might be more tired at a different time of day. No or the same amount of practice, again, is kind of linked to your dominant hand. In theory, you know, be used to catching things with your dominant hand. The weight of the ruler is allowed, or mass of the ruler, but you can't just say same ruler. We talked about this. Equipment is not something you necessarily talk about. Weight or mass of the ruler is fine. Not same ruler just on its own though. For 5C, if the question was wrong, so you didn't get 0.24 as an answer, but you could have done some of the correct working out, then you can have 1 mark. For 5D, you need the idea of student A having faster reactions, or student A catching the ruler faster than student B. You need that to be your answer to get the mark. You could have talked about the pattern overall, which was that reaction time decreased with practice, which is something else. It wasn't necessarily something we were looking at because it wasn't comparing A and B, but it is not an incorrect conclusion to get from that data. Question 6. Humans, like other mammals, reproduce sexually after making gametes in a process called meiosis. Explain how meiosis forms gametes and how these cells are different from those produced by mitosis. So there's 2 things here we need to make clear for our marks to count. We need to be able to say sort of the stages of meiosis, so what happens, but as we go, make it clear the differences between those steps and what happens in mitosis. So this is where I would brain dump a little bit about what the differences are. So 2 divisions instead of 1 division. 4 cells are then made, not 2 cells. Cells are haploid, not diploid. Remember, that means they have half the genetic information instead of all of it. And they're genetically different, they're not identical. So these are all the things we can say. The 4 cells, not 2, kind of ties into the 2 divisions, not 1, because because there's 2 divisions, it makes 4 cells. That makes sense. So they're all the things that happens in meiosis that's different from mitosis. So you can see how it's there's 2 divisions instead of 1. There's 4 cells instead of 2. The cells are haploid instead of diploid. They're genetically different. They're not identical. So I've got kind of combined statements there. So now I'm just going to turn that into sentences to make it clear what happens in meiosis and then make it clear what the differences are. So in meiosis, the cells divide twice to form 4 genetically different cells, which are haploid. There, I've got all of my stages in. So they divide twice. What do I get out of it? 4 genetically different cells that are haploid, and they are the gametes, obviously. But in mitosis, 2 genetically identical diploid cells are made. Now, I've done this as 2 statements, but I've made it very clear what's meiosis and what's mitosis. You could write it in a similar way to how we wrote our bullet points and say in meiosis there's 2 divisions, in mitosis there's 1. In meiosis it makes 4 cells, in mitosis it's 2. The cells produced by meiosis are haploid, whereas in mitosis they're diploid, and the gametes produced by meiosis are genetically different, whereas in mitosis they are identical or they are clones. You could say those statements like that and write it out a little bit long form like that. I've covered the same thing here with just writing those sentences. Sperm cells are an example of gametes made by meiosis in the testes. Explain how a sperm cell is adapted for its function. So this is just, it's actually a cells-based question, a cells adaptation question. Can you explain what specific features a sperm cell will have that allows it to do its function, and what is its function? Swimming to the egg. Okay, so I've got a diagram of a sperm cell just to kind of remind us what we're looking at. So lots of mitochondria, and I've underlined the lots of there because it's very important that we say lots, because it's about it having many or more than normal that makes it an adaptation, because all animal cells have mitochondria. But we need to explain how that helps, so it gives and provides energy for movement. Then how are we going to move? Well, we have a tail, which allows us to swim or move towards the egg. So those are the 2 main differences. We've only got 2 marks. They're the 2 main adaptations, so that is what I would talk about. So they have lots of mitochondria to release energy for movement, and a tail so that they can swim to the egg. Okay, genetic sex of human offspring is determined by the sex chromosomes carried in sperm. Complete the diagram to show how sex is inherited. 1 chromosome has been completed for you. So when we talk about biological sex, in this context, what we're talking about is male or female. And it says here that the sex, so whether an organism is male or female, is determined by the chromosomes that are in the sperm. That must mean because there's only 2 types of sex chromosome, X or Y, the sperm has 1 of each. Hopefully we know that, but there's a clue in the question there if you weren't sure. So for the sperm chromosomes, we need to put X and Y. For the egg chromosomes, we're adding another X to the X that's already there. And then we just fill the diagram in. So remember, we go across and up to look what needs to go in each box. So we've got 2 boxes which contain XX and 2 boxes which contain XY. This is correct, and we should know this is correct because it's 50-50. So 2 out of 4 boxes can give a female, which is XX, and 2 out of 4 boxes can give a male, which is XY. And that's how we know it's correct, because that's how sex is inherited. Okay, the mark scheme for question 6. The cell divides twice to form 4 gametes versus mitosis making 2 cells. You've got haploid versus diploid. You've got genetically different versus clones. So as long as your context is clear that you're talking about meiosis, then you don't need to worry too much about how you said these points. You just need to make sure that if you didn't directly compare it to mitosis each time, you need to make it clear what you're talking about is meiosis. So you need to say lots of mitochondria for the mark for 7A II, like I said, because all cells have mitochondria, so that's not an adaptation. Having lots of mitochondria, though, is an adaptation. Release or transfer energy, you can't say make energy, generate, create, produce energy, because you can't make energy, so that would not get you a mark there. So you need to say lots of mitochondria to release energy, provide, give, supply, transfer, all of that is fine. For the diagram of the chromosomes, you get 1 mark for the gamete chromosome, so that's the X and the Y underneath the sperm, and adding the X to the chromosome for the eggs, that gets you 1 mark. Then you get 1 mark for the correct boxes that you've drawn in, and that gets you the 2 marks. So you can get one of those wrong and still get 1 out of 2 marks. Question 7. A student wanted to explore how water moved through plant tissue by osmosis. They immersed cores of potato into solutions with different concentrations of sugar. She drew her results on the graph. So cores of potato kind of means like cylinders of potato, like you use a corer to get those cylinders out. Hopefully you recognise this description, which you may or may not have done with potato. But the point is that's what this question is about. It's on osmosis. So we're looking and thinking about the idea of change in mass when you put it into different solutions. And that's where our brain should be kind of going when we're thinking about these questions. So first of all they want us to do is just to add a line of best fit to the graph. People miss these questions out all the time because they don't spot them, because there's no lines. They don't realise that you need to spot. If you see 2 marks or 1 mark on the side there in brackets, if there's no lines, it means you need to do something. So if you didn't quite read it, go back and reread it so you don't miss out on easy marks. So I've drawn my line of best fit there with a straight line with a ruler. It should follow the trend, so it's clearly going down in the same kind of angle, steepness as those points. I'm hitting as many of those points as I can, or going very close to them. And I'm not extending beyond the start or the end points, which is extrapolation, because then I can lose marks. So then it says, use the graph to estimate the percentage change in mass of the if the concentration of the sugar solution was 0.4. So my concentration, or my percentage change in mass, is on my y-axis. So I need to read off a number off there. And you can see you've got the 25, 20, 50, 35. So I need to find 0.4 mole dm cubed concentration on my x-axis, and then draw a line down till it hits my line that I've drawn of best fit, and then draw it across at a right angle to the y-axis to find the number I need. So first of all, I need to work out where 0.4 is. So 0.25, and there's 5 kind of divisions in that first box. So 0.25 divided by 5 tells you what one small box is worth, which is 0.05. So then I just go along. So 0.25 add 0.05 is 0.3, then 0.35, then 0.4. So that's where my 0.4 mark is. So now I'm going to get my ruler, and it must be a ruler because you don't want to go wrong, and end up drawing a wobbly line. So draw it straight down to the line of best fit. And then when it hits that, stop, and then go across horizontally to the y-axis. And I can see that that's coming out just before 25, so it's about 24%. Luckily, 24% is the first option on my multiple choice, so I'm going to go for that one, obviously. If you didn't quite get 24%, didn't hit straight on because your line of best fit was slightly different to mine, the main thing is it's an estimate, so go for the closest option you can on that list.
Okay, so part C, give the independent and the dependent variable in this investigation. Okay, so hopefully we remember from earlier, same thing, our dairy milk ice cream rhyme or phrase to help us remember which way round is which. But also, even if we have that, then a nice way of being able to figure out always what is dependent and what is independent is using any graph or tables that you've been given in the question. So in the question, it said the immersed cores of potato into solutions with different concentrations of sugar. So they've already told us what they've changed. They've made different solutions of sugar concentration. And that is also on the graph. So the y-axis of a graph will always be the dependent variable. It will always be what was measured. And the x-axis of a graph will or should always be the independent variable. So what was changed. Because you can plot that, you will know that that's what has changed. That will also be the first column in your table is the independent, and the second column in your table will always be your dependent. It's a nice way of being able to remember, or if you're not sure which way around is which, or you can't remember the dairy milk ice cream, then you've got a graph there to help you as well. So our answer is going to be that the independent variable is the solution concentration or the sugar solution concentration, and then our dependent variable is the change in mass. Part D. So explain why the mass of the potato core in the 0.4 molar dm cube sugar solution decreased. I've clearly highlighted here the majority of this question. All it wants us to say is explain why the mass of potato in that solution decreased. So we need reasons why the mass decreased. And because of the type of practical we're talking about here and what we're looking at, hopefully we should know that our explanation has to be linked to osmosis. So we're going to have to say that the chip or the potato core lost water due to osmosis. This is the type of thing we need to write down when we're reading questions before we write our answer, because this gives us a reminder that we have to say the word osmosis in our answer. If we don't, we're not going to get marks. Then we need to explain about osmosis, because that's the reasoning that we're going to give for why the water moved that way. So remember, osmosis, water's always moving from where there's a high water concentration to a low water concentration. So in that regard, we need to be able to have that in our answer as well. So say why, explain why, our reasons why the water moved out of the potato. So we have to say where there's a high water concentration and where there's a low water concentration. So our water moved out of the potato cells by osmosis is going to be our first point. That's kind of what happened. So that's why it lost mass. So then to get our second mark, we need to explain why the water moved in that direction. So explain the process of osmosis here. So because there was a higher concentration of water in the cells than in the solution. And you can say that the other way around. So you could say because there was a lower concentration of water in the solution than in the potato cells. Either way around is fine as long as you're making it clear that water is moving out of the potato cells, so there's a higher water concentration inside the cells than outside. Okay, so part E, evaluate if the use of potatoes is a good model for investigating the movement of water via root cells or not. So evaluate questions are always a bit tricky, but we always just have to remember that's basically asking us for pros and cons, so good points and bad points, and we have to put both, at least one of each, in order to get answers depending on how many marks. We've got 3 marks here, so we need at least one of each, if not 2 of each to be safe. So it's asking us about this idea of a good model. So will it behave the same, basically? Will the water movement in these potato cores be exactly the same or a good representation of water moving in and out of root cells? So what are the similarities between the potato cores and root hair cells, and what are the differences? So how will they behave the same and how will they behave different? Because the same will be the pros and the differences are going to be the cons. So for questions like this, I would just quickly jot down some pros and cons before you write them out as statements. So both will be plant cells, so plant root cells and potato cells are both plant cells. So that's a pro because they will then behave in the same way. So link that to osmosis because that's clearly what they're asking about the movement of water. So both will have water will move by osmosis through them in the same way because they're both plant cells. Cons, differences, the potato cores are obviously thicker and wider, so they're much bigger than just individual root cells. And they're also going to have less surface area. They don't have those kind of root hairs, that shape that the root hair cells do. And obviously we know that surface area and kind of distance or diameter can affect the speed of osmosis. So that's going to be our kind of negatives. Okay, so the model is good because both cores and roots are made of plant cells or contain plant cells. And so osmosis will be the same in both. However, the potato cores are much thicker than roots. So I've got 2 pros there and a con. So I've managed to get at least one of each in, and that gets me my 3 marks. You could add one other con if you wanted to as well, if you wanted to be sure.
Okay, so let's have a look at the mark scheme for question 7. So you've got 2 marks for the line of best fit, 1 for using a straight line with a ruler and 1 for the correct positioning of the line. So there's no extrapolation, so you can't go beyond the first and last point with your line. And obviously it should be sort of similar positions to the graph on my answer sheet. For 7D, really important about wording here. So you have to say osmosis. So like we said, we have to say moved out, the water moved out by osmosis. And you have to get that to get the first mark. And for the second mark, we need to talk about there being a higher concentration of water somewhere, lower concentration of water to give that idea of a water concentration gradient. So you could talk about the higher concentration of water in the potato cells and then the lower concentration in the sugar solution. As long as you've said that and you've said that it's going from high to low water concentration, that will get you that mark. So you can say it moved out of the cells into the solution because the solution had a lower water potential than the potato cells, or you could say it moved out of the potato cells into the solution because the potato cells had a higher water concentration. As long as you've got the high to low water concentration gradient idea, you can have the second mark. And then for the last mark, we're looking at least 1 good and bad point, as we said. So you need at least 1 good point and at least 1 bad point, and then 1 other point to get the 3 marks. You've got all of those options there. Some of them we talked about. You need to make sure that you don't say that a potato is a root. It's not like a swollen root cell. It just isn't. It's made of similar tissue to the stem. So you can't say they're the same type of cell or say they're the same type of tissue because they're not. Okay, so on to question 8. So stem cells can be used to quickly produce clones of plants. Complete the sentence choosing the answers from the box. You only use each word once. So we're going to cross them off as we go to make sure we don't double up. So the first thing it says is stem cells are something. So they're not wide, narrow, or everywhere, I would say, because if you think about it, you're basically saying that means they're small, or they're large, or they're found everywhere. The undifferentiated word here is the key word we should know that's associated with stem cells. So that's the word to put there. It can become a something range of other cells. So a large range, a wide range of other cells, or a small range of other cells, or they can be cells that are all over. Everywhere doesn't make sense in here, so let's ignore that. So it's either large or small. Well, if they're undifferentiated, it means they're not specialized, so they can become any type of cell. So they can become a wide range of other cells. Makes the most sense here based on what we know about stem cells. Part B. Okay, so describe the differences between embryonic, adult animal, and plant stem cells by matching up the cell name to the correct descriptive statement. So draw one line per cell. This is the type of one where we can get in trouble if we accidentally draw two lines. If you accidentally draw two lines, make sure you've made it incredibly clear that you've crossed one out entirely to make sure that we don't lose marks here. So plant stem cells, embryonic stem cells, and adult stem cells. We need to think about what we know about these. You've got can differentiate into any cell type for the whole life, can differentiate into a limited number of cell types, or can differentiate into any cell type but for a limited time. So embryos are only around for a small amount of time, but they can differentiate into any type. So that's the bottom. Plant cells, stem cells, can differentiate into any type, and they're there the whole time the plant is alive. So that's where that one goes. And then the last one must be the adult stem cells. They're the ones that are limited, so they can only become a few types of cells, and that's why they're the middle one. This just relies on you knowing some of these facts about stem cells and being able to match it up. I would say the plant stem cells are unique in the fact that they can do it for the whole lifetime of the organism. So they're the ones to go out to look for first.
Okay, so part C. Evaluate the social, ethical, religious, and medical issues associated with the use of stem cells in medical treatment and research. You should arrive at a reasoned conclusion in your answer. Okay, so we've got this evaluate question again. So that command word, we need pros and we need cons. So we need positives and negatives, advantages, disadvantages. And because this is a 6-mark evaluate question, often we need to make sure we have a justified opinion. That's what it means by reasoned conclusion. So what do you think is best? Or what is your opinion and why? So you need that justification. And so it's most likely going to be a leveled answer, which means we need to make sure we cover all of the range of the question in our answer, and we have that justified opinion to give ourselves the best shot of getting into those top marks. So the social, ethical, religious, medical issues, sometimes these are in the question, they are clues. They're not always going to be in these types of questions, but when they are, they're helpful because it gives us some places to focus on. So the ethical and religious hints there are reasons against using embryos. We should hopefully know that there are some obviously objections to using embryos, often around the ideas of consent and embryos not or being potential human lives and them being destroyed. Medical issues, so uses, what can we use the stem cells for medical treatment, and then what are the limitations? So what are the issues medically with some stem cells? And then social normally comes under, it could come under economical, or it could sometimes be called financial. It's often to do with the cost or the availability. In this case, I'm thinking it's probably going to be worth talking about like the availability of embryos versus the availability of like donors for adult stem cells, and maybe costs or expenses that you can think about it as well, and whether everyone can get the same treatment. So these are all kind of basically, let's do kind of a bit of a brain dump. And this is what I would always suggest that you do. Not necessarily this long, but try and get as much points just kind of jotted down from your brain that you know about stem cells, because most of them are going to be applicable here. Try not to fit them directly straight into all those gaps straight away. Just try and see what we can get in terms of pros and cons, and they will naturally fill in those sections that we've been giving clues to. So first of all, what can they treat? Well, we can treat things like type 1 diabetes. We can grow new cells that produce insulin. We could treat paralysis by growing new nerve cells. Blood cancers like leukemia, when it's the red blood cells or white blood cells that are damaged. This is a pro, okay? So the pro is obviously we can treat these conditions, save people's lives, improve people's lives. That's great. Cons, so what are the medical issues here? Well, there's no guarantee that every stem cell treatment is going to work. You inject stem cells and you do hope for the best a little bit. We can't treat everything. So stem cells won't find an answer to every possible disorder that is there, but we can only treat some things. It's hard to find a donor. So if you need a stem cell donor, you can't get them from the patient themselves, then that's going to be tricky to find a match, and not very many people are willing to donate stem cells or bone marrow. It's quite hard. The list isn't very long for people who will. Adult stem cells can't treat many diseases. So if we're using adult stem cells, we can't treat everything anyway, because they can only treat a very narrow range because their differentiation ability is less. Okay, so we've got some pros and some cons there, which is good. So we've evaluated kind of the medical side of that bit for initially anyway. So then thinking of another pro, so embryonic stem cells is a pro because they can differentiate into many cell types, and so they can treat more diseases. So that's kind of like an alternate pro to the con of adult stem cells not being able to differentiate as much. And then they can also be sourced. So if you think about the fact that we talked about donors, they can be sourced from IVF treatments. So leftover or byproduct or waste product, for want of a better word, from IVF treatments. There'll be embryos that don't want to be used. So couples who have enough embryos, have enough children, don't want to use any more that they've kept in storage, or the couple's now grown too old, or they don't want to continue with the treatment. Any embryos that were created from IVF would then normally be destroyed. Or they can be donated to be used for stem cell treatment. Okay, so let's into some cons then to the embryonic stem cell option. So this is where our ethical and religious element comes in. So some people view embryos as a human life, a potential human life, and they do not think they should be destroyed. Also, as well as thinking that they have this potential human life, there's also a recognition that they cannot give consent. So unlike if you were taking adult stem cells from a patient or a donor, they have consented to give their stem cells in those cases. People that believe that for ethical reasons, obviously no consent is a problem. So these human embryos that could become human lives did not consent to be used to help save someone else's life. They don't believe they should be destroyed in this process because they have the potential to become a human. And so this is often linked to religious beliefs, as well as there's obviously an ethical element to that as well. So private treatment and storing personal stem cells. So you can get embryonic stem cells from umbilical cord blood as well, but anything where you're sort of looking at private treatment to do this, to go and get stem cells privately somehow, or you've organised your own stem cell treatment from storing umbilical cord blood, it's expensive, and so this isn't available to everyone, which then becomes a bit more of a social issue, like who is able to get this treatment and who isn't. So I'm going to organise some of these points, not all of them, but some of these points into a paragraph, which would be the style of paragraph I would consider kind of putting these together. The more you can link your ideas and show your pros and cons without it being a very obvious kind of bullet point answer, sometimes the better, because these leveled answers are looking for logical, coherent, linked points. So I'm going to try and read through the paragraph, and I'm going to try and highlight the pros in green and the cons in like a dark purple. So my pros and my cons will be highlighted so that we can see where I'm putting these points into my paragraph. Okay, so stem cells can be used to treat serious health conditions and diseases such as type 1 diabetes, improving or reversing paralysis, and leukemia. This could save or improve many people's lives. So lots of pros in that paragraph. However, adult stem cells from bone marrow, which are easy to obtain from the patient with consent, are not able to treat all of these diseases, as they can only differentiate into a few types of cell. Also, if an adult stem cell donor is needed, then it can be hard to find a suitable match. Embryonic stem cells are able to differentiate into a wide range of cells. These are therefore more useful and could treat many more diseases. They can be obtained from embryos no longer needed from IVF treatment. However, some people believe that as the embryos cannot consent and they could develop into a person, that they should not be used. Some people argue that this would be destroying a life, which is against their religious beliefs. So now I'm going to end with my justified opinion. Overall, I think that embryonic stem cells should continue to be used for medical treatment and research because they can differentiate into more cell types and can therefore help more patients with different disorders. Okay, so you can see I've got pros and I've got cons, mostly in both paragraphs. They're linked together. So I've got my however statements so that I can sort of talk about something and then say however, and then go into the cons to link those points together. So I've said some pros and then I'll say however to link into some cons that's linked to it. I've talked about both adult and embryonic stem cells. I've talked about the kind of medical uses. I've talked about those ethical and religious issues with consent, and then the idea of getting rid of lives. I've talked about the fact that it's hard to find donors, which is obviously an issue with the medical issues. There's lots more you can talk about with this. So we could have talked about, say, the idea of rejection. So if you use a patient's stem cells from their own body, like adult stem cells, then they're less likely to be rejected than if you got it from an embryo that wasn't related to them. We didn't mention the cost thing and the talk about the umbilical cord blood that you could mention as well. You don't have to mention all of these to get these 6 marks. It's about the range that you cover has got to be enough, and that you've got a logical, coherent, linked answer, and most importantly, that we have that justified, so a reason why with an opinion at the end as well. And that will get us into that top range of marks. Okay, we can have a look at the mark schemes and see what I mean by the leveled kind of descriptors there.
So the mark scheme for question 8. Mostly, absolutely fairly easy to follow. We put undifferentiated, we put wide, you get 1 mark for each. So if you got undifferentiated but you got the second one wrong, that's fine. There's 1 mark for each there. There's also 1 mark for each of those joining up with the lines. Just be careful, obviously you made it really, really clear which word you've matched up so that you don't make it unclear which one you've done, otherwise it could be ambiguous and you couldn't get marks. Okay, 8C.
We've got all of this indicative content, and I mentioned some other things as well that aren't even on here that you could talk about. And it's not a case of there is a right and wrong answer for this, as that's why it means indicative content. So it's kind of like, this is the type of thing you should be talking about. But we've got these terminology in our marked levels. So to get 1 to 2 marks, you should have covered maybe 1 or 2 of the areas. So just the idea of maybe just the religious issues or just the medical issues, 1 or 2, and without using specialist terminology. So think of the things we talked about. So differentiation, donors, embryonic, all of those kind of specialist terms that we've talked about. If you didn't have any of those in there, that would be sort of capping you at 1 to 2 marks. 3 to 4 marks would get you an answer that was linked to 2 or 3 of the areas without using specialist terminology for all of it, or which addresses only 1 in some detail. And then 5 to 6 gets you multiple areas in detail and specialist terminology, and you have that opinion with the reason. You have to get that to get into the 5 to 6 bracket. Okay? I've sort of clumped up the areas of this idea of areas of different sections in the mark scheme so you can see what we mean by areas. But like what we talked about the social idea of kind of availability of the stem cells and consent for the ethical idea, and then religious beliefs for the embryonic stem cells, and then medical issues so that we can't treat anything, and the differentiation of the different cells. All of those kind of gives you the kind of areas that we're looking at here. Okay, that is the end of our paper. Hopefully you found it useful. Hopefully you've been able to mark and get a better idea of your answers and what marks you might have got, and where maybe you're missing content or you didn't understand the question or what it was asking you, and you've managed to go through this and help you kind of realize how to maybe question, spot, figure out what the question is actually asking you for, so that then you know how to frame your answer next time. Or look at some command words and figure out whether you are explaining or are you saying enough detail in your answer to get 2 marks or 3 marks. That's what this video has hopefully helped you with. Now, because this is paper 1, obviously I suggest you go and do paper 2, and then there's another walkthrough video for paper 2 as well. Good luck.
Ouch! This is why in some videos I explain scratches.